Hi, I'm going to show you how to do a few experiments using UniProt, which is a protein database. So you can start with typing in the name of a protein that you're interested in studying. So I'm just going to put in a piece of a protein called hemoglobin beta. And you see when you type it in, you get a bunch of sequences. It'll give you an entry code, as well as the name, the entry name, the name of the protein, the name of the gene, the name of the organism, and the length, the number of amino acids. So you can scroll down here and start to select sequences that you're interested in. You can click them. You can add them to your basket, which will which you can see up here. So you can always click on your basket. So you can see if I've already added some sequences to my basket. Now we're going to talk through two techniques. I like to make sure I have my sequences in my basket and do all the techniques from the basket. But you can also do it from the list once you have actually selected some of the sequences. But the basket, I highly recommend. So let's say you are interested in doing some alignments, which is comparing sequences to each other and finding the percent identity of the amino acids. So you can select the sequences that you're interested in studying. So let's say I'm interested in this question about whether whales are more closely related to other mammals. So here are some mammals. And I'm making sure that I'm only selecting HBB. Here's another mammal. And let's say I'm also interested in comparing that whale to some fish. So here's HBB for a type of fish. Here's another HBB for a type of fish, and another HBB type of fish. So what I've done is I've selected my whale, HBB, hemoglobin beta, 3 mammals, and 3 fish. So what I can do is I can click on align, and I can go down here, align 7 sequences, and you can see that it's running the comparisons. It's taking all those sequences and comparing them to each other. Notice I have a running list of other tests that I've done in the past. So now that it's completed, I can click on the link. And it's going to do a bunch of stuff for me, which is actually pretty cool. These letters stand for amino acids. So you can see it's taking all these sequences and seeing where they're similar, where they're different from each other. But what's really interesting here is a couple of things. One is I can get a percent identity matrix, which is super helpful. So it's taking all those sequences and it's comparing them all to each other, right? So you can see that, well, for example, first of all these 100% is just comparing it to itself. So this is the cattle, bovine, HBB against itself. This 84% is showing the bovine against, I believe that's the chimp, right? Bovine against the human. Bovine against, I believe that was the whale. So you can get once you scroll over a thing, it highlights which 2 organisms are being compared, right? So that's cool. So you can get all those percent identities. You can get every combination of percent identities from this matrix. The other thing you can get is a cladogram. So you're going to click on phylogenetic tree. You see that here is a tree. I typically use the cladogram version, and it'll create a cladogram for you showing the different groups and which ones are more closely related. So you can see branching events and see groups of organisms there. So I think that's really super helpful. Now that's the alignment technique. Alignment, that's to get the percent identities and to create a cladogram. Another type of test you can do is a blast technique. So let's say you're interested in studying, and this I'm going to change. I'm going to still use HBB, but I'm going to change the organism. So let's say we're interested in using the African crocodile HBB sequence, all right? and see what other sequences are similar to it. So what other sequences are in this database and arranged by percent identities. So I'm going to click on this HBB, and now I'm going to actually, and notice I only selected one sequence. I'm going to select blast. I'm going to scroll down here, run blast, and now it's running my job. So this might take some time, but it's going to, again, create a link in which I can grab the sequences that are very similar to HBB. So here are the blast results. So again, what happened is I selected a sequence. In this case, it was the Nile crocodile HBB sequence. And I selected blast, and it is now telling me all the sequences that are similar to that sequence. So you can see that this list of sequences are typically arranged by percent identities, but it's really arranged by which sequences are the most similar. I'll explain the difference in a moment. So we can see that there's one sequence that's 100% identical. So I can actually see, well, what sequence is that? So I can click on the entry code, and it tells me, oh, it's the Nile crocodile HBB, which is not a surprise because that's the sequence that I put into blast. So I'm just going to go back and say, okay, what's the next sequence? Oh, this one is 95.2% similarity. And that's the Siamese crocodile. Mm-hmm. And then go back. And so you can see that it shows me the most similar sequences. Now, I did mention that some of the percent identities are a little wonky here. So for example, you see this sequence here is 68.8% identical to my original sequence, but it's rated higher than a sequence that's 70% identical. You would think that this one should be above here. Well, for this, the algorithm here takes not only percent identity into consideration, but also what percent of the sequences actually overlap with each other. So this one is considered more similar, even though it has a lower percent identity, because it overlaps with the original sequence at a higher degree. Not that you have to memorize that, but I just want you to sort of get this idea. But it is arranged by the sequences that are determined to be the most similar. So what's some interesting things you could do here? Well, you can identify the organisms that have more similar sequences, right? So it's not a surprise that we see other crocodiles and alligators here and other reptiles. But, if we look at some of these organisms, I wonder what organism this is here. So now I'm clicking on the organism name. And when you look at it, you notice that this is a medium ground finch. So this is a bird rather than a reptile, right? So that's showing us that, there is evidence that birds are closely related to reptiles. But something that's also interesting in some of these are some other birds. If I were to, for example, look up a reptile, I happen to know, and I have this in my cart. If I go into my cart, I see that I have a sequence. This is a reptile. What I'm going to do is I'm going to compare this to my original reptile, African Nile crocodile, HBB. And what I'm going to do is I'm going to align those sequences and get the percent identity. And once those results are ready, we'll see something really interesting. So let's click here. Let's go to our percent identity and look at that. It's only the HBB for the Nile crocodile against this tuatara is only 55% identical, right? So there are, in fact, going back to my previous jobs, and this was the blast results. Going back to my previous jobs, my previous job, that Blast, that there are birds here that have a higher percent identity than if I were to go all the way down and search for that tuatara that's going to be somewhere low in my 50s, 50 for something percent. So, in other words, there are some reptiles on this list that are not as closely related or not as similar to some of the birds that are within the top 10 similarities. So, really interesting results and two interesting techniques. So that's align to get the percent identities, and to create a cladogram to get that percent identity matrix. And then there's the blast, which gives you the most similar sequences in a database to your sequence. And those answers some really interesting but different types of evolutionary questions. All right, thank you. Take care now.